Mine's 4.
Who gets murdered by osmosis?
So it is a beautiful day. Thank you for spending your evening here. And I just wanted to share some of the work that I and the students in my lab have been doing using environmental DNA. So if at any point you have questions about anything, I would much rather have a conversation than a one-way talking session. So please feel free to interrupt at any time. I thought I would start just by giving you some idea of my background. So academia is weird in that you move around a lot. And before I was a professor, I am now at Fresno State University. I did a master's degree in Orlando, down here. And I've always been a bit of a generalist. I worked on the biggest snake in North America, indigo snakes, the most endangered bird in North America, the Florida grasshopper sparrow, and the coolest animal in North America, sea turtles. And some of that work was in Florida. I also worked on sea turtles in the Middle East, so in some really remote places where not a lot of people have been. Did my master's there. I did a PhD at Washington University in St. Louis because it was an affordable place to live. And from there I worked throughout the South Pacific, flying to remote islands and going on cruise ships, not the fun kind of cruise ships, but the sciencey cruise ships, to work on moray eels. So I've probably held more moray eels than any other person alive. Traveled to like 22 different countries, some of them not much bigger than this room, so they're tiny island nations. Also really remote places, studying a species that we just didn't know a lot about. I would love to tell you about morays if you have any questions at the end. They are super cool. After a PhD there, I moved all the way to Santa Cruz and did it's like an internship or a residency, I guess, in the medical profession, so a postdoc at UC Santa Cruz. And there I worked on the mechanics of moray eel jaws. So quick side note, because it is so cool. This is a depiction of a moray jaw where they have a normal, like, here's the upper jaw, here's the lower jaw. And this is a second set of jaws. This is actually a crowd that will know what I mean when I say the Sigourney Weaver alien style jaws.
Ooh, yeah.
When I say that to my students, they're like, oh, Sigourney Weaver. Because they're all 18. So like morays bite their prey with one set of jaws, and the second set launches out, grabs the prey, and pulls it down their throat. It's really cool. Did a postdoc studying that. Hopefully nobody has nightmares. Then I got tired of not moving to as much, so I moved all the way back to Orlando, studied sea turtles some more, studied grasshopper sparrows some more, and started studying sea level rise and climate change, which is an issue in California, but also a big issue in Florida. Then I got my first professorship at Valdosta University, which is right on the Florida-Georgia line, so I'm mostly a southern boy. There I studied sea level rise, and this is the freaky looking mouth of a hagfish, which are about this big, produce prodigious amounts of slime, have really weird freaky teeth that can also give you nightmares. Worked there for 3 years and then moved back across the country again. My wife hasn't divorced me yet, but we moved a lot. And now I study bats, environmental DNA. Why is there a picture of Edna up there?
You studied bats, it was perfect.
Nice, I planned this. Yes. When you Google pictures to put on your PowerPoint and you Google eDNA, 1,000 pictures of this come up. This is the character from The Incredibles, which I know because I have a 4-year-old whose name is Edna, eDNA.
Oh.
Everybody get it? So now I'm studying bats, environmental DNA, and amphibians and fish. And throughout my research career up to this point, I've made a career of going to really remote places and studying things that people didn't know a lot about. Then I had a kid, and going on 3-month-long cruises in the South Pacific was not much of an option anymore. So my wife and I decided that we would have research projects that were local. But what's really amazing about that is even in a place like California, where a lot of people live, even in a place that has a lot of visitorship, not as much as Yosemite, but Sequoia and Kings Canyon National Parks, we still know preciously little about a lot of the biodiversity in these places. In particular, what lives in the rivers. So So that's what sort of motivated me. And what I found really interesting about my research career is that you don't have to go to remote places where not many people have been in the past to discover new things and answer basic questions like, what lives in that river that 5 million people drive past every day entering this national park? These really basic questions. So what is eDNA, or environmental DNA? It is a way of assessing what species are present in the environment that is easier than traditional surveys. Traditional surveys are get a biologist, put them outside, and let them try to see stuff. And some of us are better at that than others. There are people who can tell you 30 species of bird just by the faintest glimmer of a wing flutter or a call. When I was doing turtle surveys, I used to be able to hear a box turtle exhale in the forest, like really well. Once I got close, I could be really quiet and hear them. Biologists are super weird. But it ends up being really hard because a lot of the species that we want to study are cryptic and hard to find and hard to see. So it's unclear if it was in the environment and you didn't see them because you can't hear a turtle exhale or something weird, or it actually wasn't there. So was it not there because you didn't see it, or did you just not see it and it was there? So if you can find the DNA of an animal, often that tells you that it is there, and it ends up being much easier than traditional survey methods. So it allows you to catch stuff, find stuff that you just don't see. It assesses long-term presence, so sometimes the DNA of species can persist in the water for months after that species has just passed through. So that allows us to detect presence of things even when it's not there for you to go and see, but it has been there recently. And because I'm at a school, Fresno State University, where we have equal weight towards research and teaching, this is a way that I can teach students, I can bring them out into the field, let them get wet and dirty in the rivers, see biodiversity up and close, but also learn some molecular biology techniques. So, eDNA, just some questions to see what you guys might know or think about eDNA. So some of the basic questions, like what DNA do we leave behind and around us? What part of your body, you're all leaving DNA in this room right now. What kinds of DNA do you think you're leaving?
Skin cells.
Skin cells, why did you go to that first?
I mean, sunburn.
Yeah.
My son was chewing my arm off.
Spit.
Spit, yep. Hair is a big one. The skin is the largest organ of your body, and it is a place where your cells shed or slough off very quickly. So different species shed their skin cells in different rates and in different ways. So what species, if you're thinking about things that live in or around rivers, what do you think would leave more skin cells in the water? What would leave less?
Fish scales.
Fish, think they'd be on the high end or on the low end?
The scales, I don't know.
Yeah, so it's a great idea. They don't shed entire scales, or they don't shed all their skin like a snake, but scales do slough off, and the scales are often continuously growing, so parts of them do slough off. So fish definitely leave DNA from their skin. What do you think leaves more, snakes or fish?
Fish.
Why snakes?
Probably because they're more hot or they're warmer and they can shed more easily.
Okay, they are just as cold-blooded as fish, so not as much as hot. Why do you think fish?
I think fish because snakes tend to shed all their skin in one go. You have a larger piece, but not
So it's
necessarily.
either hit or miss
raw.
maybe with a snake, either you get a ton of skin cells or not at all. It turns out that most of those are dead skin cells, so you're not going to get a whole lot of DNA. Same for fish scales are mostly keratin. So even your hair, it's only the root really where you get DNA, so when they pluck a hair to get your DNA. But just if you just cut your hair, there's none or very little DNA there. What other kind of species?
I think feathers, birds.
Feathers, also largely keratin. When I worked on grasshopper sparrows, you'd have to get a feather that actually had the root, much like the hair follicle, to get DNA.
How about poop?
How about what?
Poop.
Poop. What kind of, I don't want the DNA of what they're eating, I want the DNA of
Oh yeah.
the animal.
I don't know.
But sorry to get graphic, when poop goes through the digestive tract, those digestive tracts actually shed cells. How many times fast can I say more quickly than epithelial scales, more quickly than your skin. So you can get on the outer layer of poop this like sheath of the DNA of the animal that did the pooping.
Plants
Plants.
or algae?
I only study animals. but good. Yeah.
So you want like my other answer is microbes?
Yes, we also leave microbes everywhere.
Yeah.
So without getting too far off, a lot of the DNA fingerprinting that's going on in places where it really matters, like crime labs, just walking into a room, the microbiome, the little critters that live on your skin end up being unique to individuals, and you can tell just by sampling the air in a room if which individual was present in that room, if you can test that individual afterwards, which is really freaky. But let's get back to the question. What kinds of fish or amphibians or reptiles do you think leave more DNA in the water? We got fish, snakes,
Toads.
toads, all right? What do you think, they're on the high end or the low end?
Low.
Not as much. They're amphibious, so they're not in the water all the time, except when they are, when they're tadpoles, when they're in the water all the time. Okay? Yep, so as tadpoles, they leave more DNA in the water than they do as adults. Both because the adults are out of the water as much as they're in it, and for another reason that would give it away. What other kind of critters do you think?
Mammals.
What categories you threw out?
Salamanders.
Yep.
Fish, amphibians, and what?
So other types of frogs, like tree frogs, end up leaving more DNA in the water than a toad would. A lot of the newts and salamanders leave more DNA in the water than a toad or a snake, or even a fish. Why do you think?
Because they absorb water through their skin. There's more exchange between the.
Perfect. Yep. So things that have to breathe through their skin have really soft, pliable skin, and the only way to keep skin as soft as a baby's is to shed those skin cells quickly. That's not old skin. That is very new skin. And their skin is growing more quickly than it does for an adult. So things that have to go through the trouble of breathing through their skin end up sloughing skin cells much more quickly, so they leave more DNA in the water. For fish, the part of the fish, it's not the scales. What part of the fish do you think leaves DNA in the water?
Gills.
You guys went too much tonight. What do you think?
The inside of the fish?
It's sort of the inside.
The part that's like a root you're taking in?
Yeah, the same reason that soft-bodied amphibians leave more DNA in the water than toads. So, how many people have held a toad versus a tree frog? You guys don't know what you're missing. Toads end up having sort of rough, bumpy skin. It doesn't feel super wet or soft. You pick up a tree frog, it is very soft, almost like wet rubber, very soft, pliable leather, something like that. Because they're breathing through their skin a lot more than a toad does. Toads can exist in pretty dry areas. Tree frogs have to be wet all the time so they can breathe through their skin. Similarly, it's the gills of the fish where they have to exchange oxygen from the water through the gill apparatus, and those soft tissues, if you ever open up the gills of a fish, it looks like the inner guts of the fish. It's red, you can see the vascularization, looks like raw meat almost. That's where most of their skin cells are coming off of. Okay, so it's soft-bodied things that have to breathe in the water with gills or through their skin in the water that end up leaving more of themselves in the water. Things like snakes that are surrounded. Snakes are really impervious to like water pollution because they're not breathing the water, they breathe air. And they have this keratin, they have essentially a whole body covered in fingernails. Like those scales are really good at keeping water out, keeping toxins out. Also keeps water in, that's why snakes can live in dry places and frogs and toads not so much. So snakes don't leave much DNA in the water, actually it's really hard. Same for, something else that's amphibious that lives in the water but also crawls out on land?
Like otters.
Alligator.
That's true, wasn't even
Lizards?
Lizards, yep. So also don't leave much of a DNA presence, but they don't go into the water so much. I studied sea turtles, right? Yeah. So they end up spending a lot of time in the water. They'll come out of the water occasionally. We only have western pond turtles as our only aquatic turtle in California. And they breathe air, so they don't have gills that are sloughing off skin. How many people have felt the skin of a turtle? All right, more of it, that's good. Is it soft and pliable? It's pretty dry, right? So that's also skin that is very good at keeping water and stuff out, and as a consequence, it doesn't shed as often, and they're not leaving as much DNA in the water as something like a newt or an amphibian. So not all species leave the same amount of DNA in the water that they swim through. And then a basic question that we wanted to ask with this research was, is there more environmental DNA if there are more individuals of a given species present? So that sort of makes sense. If one of you can walk through this room and I can go out with a really fancy vacuum cleaner or something and suck up all the skin cells that might have fallen off of you as you walk through the room, and then I can maybe detect whether or not you walk through the room or not. If 100 of you walk through the room, I'd probably be more likely to detect you than if only one of you walked through the room. So in the project that I'm about to describe, we took environmental DNA samples. We took water samples and tested for the DNA of species we expected to be in the rivers. But I also took a horde of students with me, and we did sort of play peekaboo. Another baby wanted some peekaboo. In the rivers where sometimes the water was as deep as this room is tall, where we're snorkeling through, and at other times we were army walking in 4 inches of water, sort of on hands and knees, just poking our head in and looking to see what species of fish were in this much water. And if we saw more of them, we were expecting to see more presence of their DNA in the water. So that's sort of our working hypothesis.
That's the question.
Yes.
So you're making a big deal about how much DNA is going around. How much do you really need? You know, if there's one dead tree out here or 1,000, it doesn't matter. I can see the one dead tree.
That is a great question. So DNA is too small to see, so it's very, very tiny. Molecular biologists have gotten really good at detecting it at very low quantities, but we still often need a decent amount of tissue. So if I were to go do a genetic study of otters, I would need a piece of tissue that's probably about the size of a pencil eraser, something like that. When you get a biopsy done for if you have a lump somewhere or something you want to test it, they take a chunk, maybe a little smaller than that, but something that you can see. You know, so sampling water, where maybe there's 1 or 2 cells that I have to have a microscope to even see, and each one of those cells might have some DNA that's too small for me to see with a microscope, you end up needing a decent amount to be able to test them using the methods that I'll describe. Great question. So, the way environmental DNA projects work is a little different than you might imagine. It's not like we take a bit of water and then we sequence all of the DNA in it. To do DNA sequencing, you need a decent amount of source of tissue, of DNA. In this case, it's a little more complicated than that, where you only find what you're looking for. So I was trying to think of an analogy that worked, so try this. Imagine the shirts that you are wearing are a part of you, and when you reproduce, you pass on, the same way you pass on your genes, your offspring are going to be born with the same color shirt that you have, or some amalgam of you and your partner. Everybody with me on the analogy? All right. And your shirt is going to shed threads as you move about and go through your life. And I might be able to detect whether or not you are present in a room just by the threads. If I find a dark blue, I might know you were there. If I find something black, I might know you're the only one wearing a black shirt. You must have been there. But I can't just, the threads are so small you can't see them. So what we do is we say, well, I know that you guys are the candidates. This is a fixed number of candidates that might be present in the room. So I'm going to make what we call a probe, a sample from each of you. I'm going to go to each of you, I'm going to pluck a thread out of your shirt, and I'm going to make a library where I have one thread for each person, and I know which person matches with each thread. So that sort of Sherlock Holmes style, when I find a thread in the environment, I can match it to that library. That's really powerful because if I find one that matches, I know who it came from. It's not powerful because if someone new comes into the room, I got nothing. I have a thread that doesn't match, I don't know what that is. So there might have been many other things in our sampling that that were present that we didn't find because we weren't looking for it. I didn't have a reference library. Does that analogy make sense? Everybody with me? Cool. Okay. Because we want to know what to look for, we have to get into the water. I'm a big fan of field work. I like taking students in the field, so I wanted that excuse to be able to pluck them into freezing cold rivers and do fish surveys with mask and snorkel. So, this work occurred in Sequoia and Kings Canyon National Parks, which are right about here, so just all of California. We zoomed in. This is Sequoia National Park, this is Kings Canyon. Notice this little square part. This right here is, the town of Three Rivers is right about here. This is Lake Cahuilla. Zooming in, here's that square part. Here's the lake. And zooming in even further, here's the lake again. And it's made up of three main tributaries. The upper is the name of Three Rivers, also the name of a very good brewery there, if you haven't been. Sometimes called the north, middle, and south, or let's go with that. The north, the middle, and the south fork of the Cahuilla rivers. So our project sampled all three of those forks, and our study design was to sample inside of the national park in each of the rivers, and then outside of the national park in each of the rivers. Under this basic idea that outside of the national park, here's how the fieldwork would go. When we were initially scouting out places, we'd go inside the park, it would be beautiful, pristine, quite lovely. We'd go outside of the park, there were diapers washing down the river, there was McDonald's bags everywhere. Everyone and their brother goes and does whatever they want to do along the river outside of the park boundaries. And that happens sometimes inside of the park too, but much less. So we sort of went under the supposition that biodiversity should be higher inside of the park than it is outside of the park. So we'll see how that plays out. So we would go to a sampling site, so there's 6 sites, the 2 sites in the upper, inside the park, outside the park, in the middle, inside the park, outside the park, in the lower, inside the park, outside the park. And at each site, we would split the river into 4 sections, so I'd have to climb over rocks through the river with a measuring tape and measure out 25 meters. If the water was flowing this way, we'd take a water sample right here, and then we'd get in the water and we'd survey in this first quadrant everything we'd see and how many of them we would see. Then we'd survey the next one, which species did you see and how many of each species, every bit of water. So sometimes we'd have 6 people moving in a line with mask and snorkel, sort of just to count everything for all of these. And the reason we split it up like that is because I would suspect that if I see 1,000 of fish species X, I'll probably get more of their DNA than if I saw 10 of them. Also, if I saw fish species X here, I'll probably get more of its DNA than if I only saw it here, because I'm closer to the source. Does that make sense? Okay. So, it's kind of dark. This is the National Park Service biologist who initially envisioned the study and who we worked with, Eric Meyer. This is a very cold colleague of mine. We were doing this up into October, up at like 5,000 feet in the river, so it was really, really cold. Me and one of my master's students. And in some of the larger parts, it looked like this, where we're snorkeling in fairly deep water, sort of moving across in a line and counting everything we see. So this is at, anybody recognize this one?
Thank you.
This is at Potwisha. Often we'd have visitors. And this is a really nice picture of one of the upper Cahuilla sites where this water is like 3 inches deep everywhere we went. Sorry, the image is a little bit grainy. But in this case, you sort of have to span out and you would just crawl and stick your face in 2 inches of water, and you'd be surprised how many fish get freaked out when a giant head comes into a tiny little pool this big and they swim around. But it makes them easy to count because they all jump when you stick your head in. So we go to each site. We did this over the course of a full summer and into the fall, sampling every site about 6 times. Oops. So me coming up with plans, taking students out. And after we take our water sample, we would immediately take that up to my vehicle, and we'd lay that out with a a vacuum pump and a filter, a piece of filter paper. So we take 2 liters of water and we pour that water through a filter about this big that would collect anything that's organic. So we would make sure not to get any silt or anything in it, and otherwise every piece of the useless stuff, plants and algae and things that aren't that interesting. So any piece of plant matter, but also any cells that came from an animal would also be in that water sample, would get caught up in that filter. And then we'd filter 2 liters of water through, and then we'd take that filter back to the lab, and that's what we would use to isolate DNA from. Some of the things that we were looking for were animals that we could see. I'll show you some pictures of some of the animals that we were looking for, but we were also looking for this really detrimental disease. These are all dead frogs, if you can't tell, that has been decimating amphibian populations all over the world since the Korean War. Anybody know what this is? Yep. So it's often referred to as BD, as an abbreviation, or chytrid fungus. It's a fungus that came out of South Korea, probably came back on the boots of a soldier or someone from the military, and has since spread all over the world. And it grows in the skin of amphibians and makes their skin, the fancy word is become sclerotinized, or it's become hard and crusty. And that's not so bad if you're a snake. That's not so bad if you're a lizard. You have hard, crusty skin anyway. But if you have to breathe through your skin, hard and crusty doesn't work. And you suffocate and die. So it's really detrimental to amphibians, and it's been knocking out amphibian species all over the planet.
What is the disease called again?
BD, like B as in boy, and then D as in dog.
Does it affect people?
It does not, but it's very easy. So it's very easy to spread because it's a fungus that can persist as spores. So they could have been on someone's boots for years in their basement, and then they took those boots out, stepped in a river, the spores germinated, and it spread. So every single time we would go into a river and do our samples, all the students would have to line up. I'd have a 5-gallon bucket of bleach and put like diluted bleach in, and then students cover your face, and I would throw a bucket of bleach on the front of them, and throw a bucket of bleach on the back of them, and next in a line. To make sure that we sterilize them. It was like less than 10% bleach. No students were harmed in that. Totally fine. But that's been shown to knock out the fungus so that we're not inadvertently spreading it by doing these surveys. Which by the way, there has never in the history of the park been a systematic survey of what fish and amphibian species are in those 3 rivers. That's where the largest organism on the planet is. We're like 10 miles away from it, from the General Sherman tree. Like lots and lots and lots of people go there. The national park is, I don't know how old, more than 50 years old there. We've had a presence, we've had biologists there for many, many decades. We don't know just the very basics which species live in that river. So we were looking to see if this fungus was there. So we had a library of the DNA for BD to see if it was present. We're also looking for some of these species. What's this one?
Bullfrog?
Bullfrog. Shouldn't be there. Bullfrogs were introduced largely because they were used as bait for fishing. They are now everywhere. They eat turtles, they eat birds, they eat snakes. They are voracious predators. They get to be like about this big. They're native where I'm from in the southeast, but they should not be here, and they are here. So we were looking for them as an invasive species because they're really bad for lots of the other species that are present. Anybody know this one?
Scorpion.
Nice. Because of the beer with the name, or how do you mean it? This is the riffle sculpin. What about this one?
Brown trout.
Brown trout.
And a rainbow.
Rainbow trout. And this one's not a bullfrog. What's this one? It should be here.
Yellow leg.
The yellow-legged mountain frog or yellow-legged frog.
You get a prize for the night.
Misfit youth.
Nothing wrong with that. Well spent, yeah. We talked about this one already.
That's a toad.
So you have, this is just the western toad. You have your Yosemite toad here that's unique. What's this one? He's holding his head up to show his,
That's a salamander.
That's the California newt. Yep. So this one's the one that's super toxic. And it's advertising the fact that, like, hey, I'm really unpalatable. You definitely don't want to eat me by having those bright colors. And when they're harassed, they'll show those bright colors. There's two ways to be poisonous. You can, well, if you eat me, you'll experience the poison. Maybe you won't eat my brother. But that sucks for you because you still have to get eaten. Or you can just have really bright colors to say, like, you really don't want to do this. I'm not afraid. I don't need to hide. I have bright colors so everyone can see. And what it should be. Right, so the California newt. You might know this one. It gets to be about that big, pretty aggressive.
Is that a squawfish?
Nope. Oh, well, we called it the pike minnow. That's its common name. I haven't heard of squawfish. Same thing. So this is a pike minnow. And this is a baby western pond turtle. They are. Bullfrogs eat them. So these are those species. Each of them is listed here. And if there's an X, that means we detected them using environmental DNA. So it's actually really cool. When I say we made a library, essentially it's a tiny piece of glass. And on that piece of glass, everybody remember what DNA is shaped like? Yep, so it looks like a ladder. So it's called double-stranded, where you have matching nucleotides on each rung of the ladder. So it's 2 rungs with things connecting them in the middle. You can take that DNA and if you take off one of those rungs, you can always recreate it because there's only 4 nucleotides in DNA, and if you know that one is, these names are not important, is an A, you know that A always goes with T. So I don't need the other rung of the ladder. If it breaks off, I can rebuild it. I know, because if I have one rung, I know what's complementary to that. Does that make sense? Not reaching too far back into your biology training? That ends up being really powerful in making this library like the library of strings from your shirt. So I take this little piece of glass, I go find a pike minnow, I go find a western pond turtle, I go find these species, I take a little bit of their DNA, I denature it, that means rip off one of the rungs of the ladder, and I tie those ladders down to that piece of glass. So now I have a piece of glass with half of a piece of DNA from each species, and each of them have a unique piece of DNA. Just like each of you would have given me a unique thread for your shirt. Everybody with me? You can blast the laser onto that, and the laser will only make it glow if it's a full ladder of DNA. Half a ladder of DNA, no glow. So when I have my blank little glass sheet, I shine the laser on it, nothing glows. I take my DNA that I isolated from the water and I wash that across the sheet. Anytime I had a piece of DNA that came from a western pond turtle, that piece of DNA is going to wash across that sheet, bind to the other half of the ladder that it matched, and now I'll have a full ladder. I'll have double-stranded DNA. Does that make sense?
It does that on its own?
Yes.
Wow.
So it's like Velcro, very much like Velcro. So if I put the DNA that matches something that's on my reference library, my little piece of glass, as soon as it finds its mate, it'll stop and bind there. And then I can shine my laser across. It's like a grid where I've got a little piece of DNA in each cell. And this cell might be the western pond turtle, and this cell might be the pike minnow, and this cell might be the western toad. When I shine my laser across, nothing glows. When I wash the DNA across, and let's say a piece of DNA that was isolated matched here, and another one matched here, and another one matched here, I'm going to see a light shine up on this cell, and I can look and say, the top left cell, ooh, there must have been a pike minnow in that water sample. Oh, and this one down here, that must have been a tree frog or whatever species I was looking for. So it ends up being really easy once you do the molecular work to survey which species were present or absent because DNA acts in this sort of Velcro fashion. Where if you have a library, you know what you're looking for. When you put your sample across that, it'll automatically bind where it matches.
Those
sample, not the samples, but the slide reusable? Or do you have to-
It is reusable. Yep.
You don't have to come up with a new one every time you want to do a DNA test?
Nope, they don't last forever, but you can reuse them a couple of times. DNA is like Velcro, and the way that you rip the Velcro off is to just heat it up a little bit. So because these pieces of string are tied to the glass plate, I can just heat it up, and all these will wash away again. And I can wash a new sample across it. It gets old if you do it too many times. But it's super cool. Okay, so if there's an X, like brown trout, upper North Fork. Upper always means inside of the park. Lower means outside of the park. So these are both North Fork. I got the brown trout. Environmental DNA found it, but we never saw it doing our visual surveys. Rainbow trout. Environmental DNA says it's inside the park, outside the park, but I never saw it. No DNA and I didn't see it. Yes DNA and I did see it. eDNA said the pond turtle was there and I saw it. eDNA said the pond turtle was there but I didn't see it. Bullfrogs saw and detected. Western toad, I saw them there, but we didn't detect it with eDNA. And what do you think I saw of the western toad if there was no eDNA? A tadpole or an adult toad?
Adult.
Nice, you guys are good. Because they don't leave as many skin cells in the water. Yep, very good.
So how did you know to use brown trout if you never saw any? You just speculated they might be there?
I knew brown trout were in those rivers, so I made them in the library, right? There might have been 15 other species of fish present there that I didn't see, but that had DNA in the water, but because they weren't in my library, you only find what you're looking for. So, there's only a couple places where I know it was there because I saw it with my eyes, but I did not detect it with the DNA. And in every case, that was when I saw them in the furthest, so remember those 4 quadrants? Like, I took the water sample and then close, further away, further away, further away. In both of those cases, I only saw a couple of them, individuals, and they were as far away from where I took the water sample as they could be. Which sort of makes sense. The closer I am to you, the more likely I'm going to pick up one of the fibers from your shirt. The closer I am to you, the more likely I'm going to pick up some of your environmental DNA. But generally, the pattern that should come out from this is that 1, scientists make really ugly figures, sorry. 2, if I saw it with the environmental DNA, I also saw it with my eyeballs. There's only a couple places where environmental DNA didn't pick it up, and they were only a couple individuals when that happened, which sort of makes sense. But there are lots of examples where environmental DNA told me it was there, and is pretty foolproof. There's no other way for that thing to light up with the laser unless its DNA is there. But I didn't see it. Which ends up being really powerful because historically the way we survey rivers is to do what we did. You get in the water and see what you see. And most of the time, you're picking up maybe half of what's actually there. Because lots of things are hiding under rocks, or they go away as soon as you get in the water, or whatever. So environmental DNA allows you to pick up things that you just wouldn't see otherwise. Which of these is harder to catch, brown trout or rainbow trout? Really?
Which ones are harder to catch?
Yeah.
Are you fly fishing?
I was curious because I ended up finding it harder to find brown trout. When I did find them, they were always hiding under a rock. That might not mean have anything to do with their affinity for a lure. But
So you use bait.
yeah. But the rainbow trout were always out in the open. You could see them from the shore. So, eDNA is likely foolproof. There's pretty much no other way for that little reference plate to light up when I blast it with the laser unless the eDNA was actually there. Things that are not really abundant in the water, or something like a toad or a turtle that's not leaving a lot of skin cells in the water, is unlikely to get picked up. So I might see it and know it's there, but not get its environmental DNA. There are other things that might be common, but don't leave eDNA, largely like a snake. There were several times, I didn't list them on here, where we caught snakes in the water and we added them to our library, so I know they were there, but they didn't come up, and that's because they have those skin cells, they're covered in fingernails. Like, they aren't sloughing skin cells that have DNA in them. So it ends up being useful to do both, to take water samples, do environmental DNA, but there's also things that you can't pick up with environmental DNA. So we do need people to get in the water and get dirty, and that ends up being more fun anyway. Otherwise, you just drive there, take a water sample, and go home. So yeah. The other thing that ended up being interesting is that, so inside the park, outside the park, fortunately no BD, except outside the park at one of the sites. But at least one species that's not present here. Here, at least one species that's not present here. Here, another species, although it's invasive, it's not present. We ended up seeing more animals and one or two more species outside of the park than we saw inside of the park. So the McDonald's bags and the dirty diapers and stuff that were left, these areas that end up being more polluted, ended up having as high or higher biodiversity than the parts that were inside of the park. We could talk about that at the end, but that was sort of surprising. So I think I'll end there, if we're okay on time. Yes, plenty of time. If you want, I can talk about western pond turtles. We do some genetics with them that's kind of interesting. Or if you guys have questions about what I presented to you, we can talk about that.
Is your DNA the same as mine? In other words, if you put me on the slide, would you light it up?
Great question. So, it depends. If I wanted to find out if Jerry the sculpin was present in the river, but Suzanne the sculpin was not present in the river, I would choose a different part of the DNA to make my library than the part that I chose. The part that I chose distinguishes any sculpin from any other fish. So our DNA is 99% identical. Very little of our DNA is different from each other. But there are definitely parts that are different from each other. And if I wanted to know if you or I were present in the room, or a chimp or a dog was present in the room, I could choose many different markers that would distinguish. But if I wanted to distinguish between you and I would choose a very particular subset of DNA to look for. So when I say I take a piece of their DNA and attach it to this, I'm taking a piece of DNA that's maybe 100 nucleotides long. Those A's and T's and G's and C's that make up your DNA, maybe 100 of those. Your genome is several hundreds of millions of those. So I'm taking a very tiny, very targeted, non-random piece of their DNA, a very particular gene that allows me to distinguish one species from another.
So
I couldn't distinguish between individuals of the same species.
So you use CRISPR or something like that?
So CRISPR is a tool for editing DNA to change it in a way that you want it to be changed in particular.
Not for cutting it?
No. It does cut DNA, but in this case, what I do, you use a you use an enzyme that cuts DNA in a very particular place, much like CRISPR is targeted, to get this little piece of DNA that I want. Great.
Second question.
Yeah.
Sorry, second question. What's it feel like to get bit by a moray?
I have never been bit, that's why I have 10 fingers. There are a couple other people who work on morays, and they all look like this.
Yeah.
Really?
Yeah, they have incredibly sharp teeth, really strong jaws, and they're just super mean. So I do not know, but only because I've been very careful.
So if you do get bit, does the second jaw suck you in?
So they use it to swallow, since you asked. All right. How do you guys swallow your food? I know how you get it off the fork, use your teeth.
With your tongue.
Your tongue doesn't push it down your throat.
Pulls it back.
Saliva.
Maybe to the back of your mouth, but how do you get it all the way down here?
Swallow.
The throat.
That's the traction muscles in your throat.
There's some peristalsis that helps a little bit, but that doesn't do it.
Gravity?
Gravity, yep. We are all gravity feeders. We catch food with our mouth or with our hands, but we get it into our stomach using gravity. Virtually every terrestrial animal uses gravity, not all, but most. How do fish swallow? Their stomach is horizontal with their mouth, so it ain't gravity.
Water flow.
So you have to like swim faster to swallow? It is water flow,
Okay.
but not by swimming faster.
Respiration.
What is it? Were we going
Fish?
fish?
Water.
Huh?
Fish?
Yeah, how does fish swallow?
They open
For
their mouth.
gills?
Yeah, so what does a goldfish look like?
Yeah.
Those plates on the side of their mouth, they're called operculum, are flapping, and that's where the gills are behind. And when they flap like that, that's creating a vacuum and moving water over the gills. You've probably caught a largemouth bass, and when you pick it up, what's the most prominent feature?
Like you mean mouth?
Yeah, it's huge, right? Why do largemouth bass have enormous mouths?
They create a vacuum.
They create a vacuum. So largemouth bass and lots of predatory fish rarely ever touch their prey with their mouth. Often they go straight to their stomach. So you get close, they open their mouth to this enormous volume, it creates suction, food goes right in. They close their mouth, and if you watch them swallow, they just open their operculum a few more times to sort of wash the water back and push the food into their stomach. To do that, if you've ever watched a slow-mo video, you can find them on YouTube, they rapidly expand their operculum so that, if you're looking at a fish, they're fairly narrow-bodied, right? But when they do that, their gills make them very wide-bodied to create that big vacuum. Morays are super cool because they feed inside of the coral reef. When you go out on a coral reef and a big grouper or something comes out, all the little fish go into the reef because the grouper can't go in there. But morays can. They're very skinny and narrow-bodied like a snake, so they can go into those holes just fine. So they've evolved to be long and skinny, but they don't have that operculum to expand because they're in a tight space. They can't do it. So they can catch the fish with their jaws, but they have a problem. They can't swallow because they can't open they don't even have an operculum. They just have a little hole in the side of their head. So because they can't do that, they have this second set of, they're called raptorial pharyngeal jaws. Raptorial because they reach out and grab. So this second set of jaws mechanically ratchets the food down. So it's not to bite you twice, even though that's what it does. It's to swallow, because they can't use gravity, and they can't use suction like every other fish.
If you're snorkeling or scuba diving, say a couple feet away from a moray eel in a little cave or wherever he is he likely to come after you, or do you have to tempt him?
It depends. Some species are just mean, and they will, but most just want to be left alone. If you're spearfishing, they'll come out and take the fish off of your spear. They're aggressive.
Do other species have this same
Morays are the only that have pharyngeal jaws that do that.
Is there any other species that you know have like really weird jaws, or like just in general?
Well, lots of other fish have jaws that they have a second set of jaws in their throat, but it's usually for grinding their food up. So like sheep's head, things that eat clams and stuff, will bite those clams off, but they don't chew them and masticate them with that mouth. They put them in their throat, and then their throat jaw grinds them up, and that gets them into small pieces of your stomach.
So what's the Park Service going to do with this chart that you showed us?
Oh, yes, great question. Thank you for circling back to eDNA. So this provides a couple of things. One, it tells us which species are where, which is really basic information that you might think, okay, who cares, whatever. For biodiversity and for conservation, you can only conserve what you know about. And if things started going extinct, if we started losing amphibian or fish species, if we didn't know they were there to begin with, we wouldn't know that they went extinct. They would just disappear in a vacuum. So this sort of data, baseline data, is really crucial just for monitoring, just to know what's out there and how's it doing. 10 years later when we do this, are we going to get the same species or not? The other thing that makes this really useful, especially for me for teaching classes, is that there's more species outside of the park in less pristine habitats than there is inside of the park in very pristine habitats. And that ends up being a tool for teaching this thing called, that relates to the importance of disturbance. So there's this thing in biology called the intermediate disturbance hypothesis, which is this idea that biodiversity is highest when you have just the right amount, not too hot, not too cold, the Goldilocks effect of stuff getting messed up. If you perturb the environment, you disturb it too much, that's bad. If you don't disturb it at all, that's bad. But if you disturb it just enough, that's good. And the reason for that is, species usually take one of two strategies. You can say, I'm going to be a really good competitor. I'm going to take my time, I'm going to set up house, and I'm going to defend my territory, and I'm going to outcompete everybody else who's there. That's one strategy. Another strategy is to say, competition is hard, not doing it. I'm going to go wherever nobody else lives. As soon as a tree falls in the forest and there's an open space, I'm going to be the first one there. Once other stuff comes, I'm a terrible competitor. I'm going to get outcompeted every time. But that's fine, because I'm always the first person to a place that gets wiped clean, that gets disturbed. Right after a fire, right after a tree fall, right after a thunderstorm that washes away soil, whatever it is. And if you have no disturbance whatsoever, you only get species that are good competitors. They outcompete everybody else, and there's only them. If you're in a place that gets disturbed all the time, you only get these species that are good at getting there first. You don't get any of the good competitors because they take too long to set up shop. As soon as they get there, it gets disturbed, they get wiped out, and they come back. Right in the middle is when you have both. They can both coexist. And a lot of the management practices that Park Service uses is, and you see this especially in Yosemite, is let's manage to make it look like Ansel Adams photographs. Like, nothing can change, it needs to look exactly like that, that's exactly what it needs to look like. No, we can't let anything else happen. And it's complicated, but sometimes we err on the side of not having enough disturbance by trying to manage things to be what we think they should look like. And if you don't have enough disturbance, you're getting away from that Goldilocks amount. So we don't need dirty diapers and McDonald's bags to maximize biodiversity, but things like putting in bridges, having some houses, other manipulations to the stream might be that amount of disturbance that would have occurred naturally through tree falls and fires and mudslides and things that we've sort of worked to minimize in lots of other ways, because management strategies in national parks are very complicated. Nothing but respect for those people who do that. But often they are pushed to manage for this ethereal idea that the public wants, not necessarily what is natural for that system. So I think that's what explains the difference in biodiversity.
That was a lot of work to get just a little bit of data in one fairly short area of one river.
Yeah.
I mean, if you look around, I mean, you've got a lifetime of work here and a lot more.
Yeah, it ends up, we did some spot checking elsewhere and you ended up getting the same suite of species. So that's one of the ways you know if your survey is worthless or not is, let me go repeat that in one other place. Do I get the same thing? Okay. Let me repeat it in another place. Do I get the same thing? Okay. Let me repeat it in a fourth place. Do I get the same thing? Pretty much? All right. I probably only need to do one or two. If the second time I repeat, I got a totally different answer, then one is not enough. And in this case, we haven't surveyed the whole river, but in our other sort of spot checking, it looks to be like a representative sample. But you don't know until you do everything.
So you think it's representative of the river out here?
I think it's representative of the Sequoia. I have no idea. I think it's a totally different river system. And you get much more pulse of high water in the rivers here than you do up there. You get snow melts and you get raging rivers, but nothing like you do here. Nothing like the Merced.
What do you plan to look at next?
I like this, but I've been doing bat acoustic research in Yosemite here and in Seki, where we try to see which species of bat are present by putting out recorders that listen for them. And you can tell each species of bat makes a unique call, much like they leave DNA that's unique. And you can tell which species they are by their calls.
So what do we have?
Sorry.
What do we have here?
Yep, so right now there are about 13 species of bats that are pretty common in Seki and Yosemite. In the valley is where my recorder is now, and it's just been too cold. They're super active in August and September, and not much the rest of the year. And again, we don't know anything. Do they leave? We know that they hibernate. Do they hibernate most of the year in Yosemite but not elsewhere? Do they fly into the valley where it's nice and warm? Nobody knows. So really basic questions. And bats are the most diverse group of mammals. They eat mosquitoes, people like that. They eat crop pests, like they're a really important component of biodiversity, but they fly around at night, they're hard to see, so we just don't know anything about them.
So what frequency are the bat calls, and how do you tell one from other, by frequency or by pattern?
Both. Most of them call in the ultrasonic range above 20,000 megahertz. That's usually where human hearing is. Some of us
Kilohertz or megahertz?
kilohertz. Some of us can hear up to 25 or so. So I guess it could be 20 kilohertz. Yeah, not 20, 20 kilohertz. So up to 100 is where they call, and both the frequency that they call at and it's called a sonogram, but it's essentially the word. I mean, they're using their vocal cords the same way we do. So some bats echolocate by saying like click, 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 and some echolocate by saying click, click, click. They're just different syllables of the same word, so to speak. And you use a fancy computer algorithm to distinguish, but you can visually tell most of them apart.
So are they communicating or just echolocating?
Some species nest in big groups, and this is also cool. When you're social, you end up having to have the need for more words. So if you need to interact with these people, you're gonna have to develop words like, hey, that's my food, could you please move, or let's mate, whatever, like things that you need to be able to communicate. If you're solo, you pretty much just need echolocation to find food, and mate, that's it. So they have 2 words, except for the social ones. They often have 20, 30 words and are very communicative. Our bats are not social bats. Those are mostly like fruit bats. So they just have 2 words, eat babies.
If you wanted to know every species inside a river, how would you go about that? Because libraries only have
Yep.
so many DNA,
Yeah.
samples, so.
You essentially can't. You could sequence all the DNA in that sample, but you wouldn't know which DNA goes to what. Are you getting the bacteria? Are you getting the algae? Are you getting other species of fish? So you'd have to catch everything that's there, which you could do, sequence its DNA, and then build a bigger library, essentially. We started small because we knew these were things that we already had DNA for, and that should be present in those rivers.
Do you think there's like a lot of species that just aren't known?
We probably know the species that are there, but there's certainly some species that are at low abundance that we just don't see and haven't picked up because we didn't know them before. But I doubt there's like 6 more species of sculpin here that we just haven't discovered yet. We've probably found them all. But which ones are present there? There's probably more than what we see.
That really depends on the group, right? Like-
Yeah, some groups have cryptic species, like butterflies are really known, fungi. Virtually every fungus you sequence the DNA of is a new species. But for fish, for birds, for amphibians, for reptiles, it's rare that we would discover a new species in North America. It still happens. But for those, we pretty much know
Okay, so before when you were saying when someone walks through a room because of their unique
microbiome. Yep.
microbiome that builds up on them. Let's say you're a twin. Would your microbiome be very similar, or is it really just very unique to the person?
Great question. So it's both. Your microbiome is determined by your genetics, in small part, but it's also determined by your environment, mostly by your environment. So if your twin lived somewhere else, lived in Kansas and came here, you guys are likely to have very different microbiomes. But some of them might be similar because maybe you have some genetic abnormality or oddity whereby bacteria X just loves your skin and grows really well in your skin and nobody else's because they don't have that DNA. In which case, you and your twin might have some similarities, but it's mostly the environment we're from. So we each have a unique microbiome on our skin and our guts because we eat different foods, because we have different genetics. But if 2 of you live together, share the same, towels, clothes, washing stuff, and you eat the same food, you're likely to have similar microbiomes, but not identical, because part of that is determined by your genetics as well.
So I know some identical twins don't always they're not the same. You know what I mean? Do you know the reason for that? Why
There's
are
lots of types of twins. You can be genetically identical.
Yeah.
You can be fraternal twins, or where half of your DNA is identical. And then even when your DNA is perfectly identical, there are dimensions of your development where certain genes get turned on early in life and stay on, and that might not be the case for your twin. For your mom ate something spicy and it affected one of you more than the other, like for things we just barely understand. Feel free to chime in. But yeah, so you might have the same DNA in your cells, but it might be turned off on you and on in your twin. And that can explain a lot of differences. We figured that out with mice, where there's these cool studies where you take isogenic mice, genetically identical mice, raise one with a bad mother and raise one with a good mother, and then raise those babies with a good mother for like 10 generations. You'll still get differences 10 generations later between them because of a bad great-great-great-great-great-great-grandmother. Something about that bad great-great-great-great-grandmother turned off certain genes that stayed turned off for generations. DNA is exactly identical, but they're still behaviorally different. That shouldn't happen, but it totally does.
You know what happens in my family?
Be a good parent. Well, thank you guys for your attention. I'll be around if you have questions.